I'll start by, I'm talking about the PanCare Life project here. And this was another complicated data collection exercise. This is the second project that PanCare undertook. It was coordinated by Peter Katch out of UMC Mainz, and the management team consisted of Desiree, myself, Helen Campbell, and Kylie O'Brien from Ireland, with over 100 consortium members. Project ran for 5 years, from November 2013 to October 2018, and the EU contribution was 6 million. So as we said earlier, 6 million doesn't go that far when you've got 5 years and you have 100 project members. But many of these projects members shared their data at no cost to the project, and I really am grateful to them for their generosity. PanCare Life dealt with serious but not life-threatening late effects, and it's more of a clinical than an epidemiological approach. We were responding to an EU call for investigator-driven supportive clinical trials and observational studies. And this was a very broad call, but we were successful in getting ourselves funded. So you might be asking yourself, how in the world could we ever assemble these projects, and how could you be part of these projects? Well, it's a lot of talking, I'll say that. It's a lot of meetings during the PanCare meetings, lunch, breakfast, evening, many telephone calls. And when we met at other places like in Atlanta, Georgia, for the CCSS meetings and the Dan Green meetings, we also had meetings to develop our PanCare projects. So we took the opportunities whenever possible to have these discussions, which resulted in defining the topics that would be included, defining who would be included in these projects, and people included themselves, and they also dropped out for various reasons. So it's a lot of trust, and it's a lot of discussion, a lot of toing and froing, and we really didn't know at the beginning what we were going to get at the end of these discussions. It did take a long time. It took maybe 2 years for PanCare Life to take shape. So I would say there are a couple of basic requirements. The leadership team should have strong institutional support, and the institution should have a track record with EU projects, by which I mean that they should have experienced people in financial, legal, and research offices. This really helps. And the team should include experienced study managers and research managers, and in particular, the administrators of the project. And we had Pintail as have other projects, and they're just terrific. It makes a huge difference to be able to hand off to somebody who knows what they're doing, all the administrative details of these projects. So we had a 3-part leadership team with the coordinator, the research manager, and the administrative manager. And that worked very well. We had very frequent meetings, both in person and on the telephone, over the course of the project. The project, PanCare Life, stated that we would use observational and molecular genetic investigations to study late effects that impact fertility, hearing impairment, and will assess health-related quality of life. The information will be incorporated into new guidelines for fertility preservation. We had 17 beneficiaries and data providers from these countries, and the following countries below were also involved as data providers. They were not initially beneficiaries. Just to say a bit about those 5 data providers below, we in some senses solicited their involvement because they brought various strengths with them. So they didn't propose themselves at the beginning, but we went and looked for them, for some of them at any rate. Additional cooperating partners included the Audiological Reference Center in Münster in Germany, 2 genetic labs in the Netherlands and 1 in Germany, and the very important Ethics Advisory Board. We spent a long time on ethics discussions, and they were really very, very helpful. The entire data pool in PanCare Life contains data from 32,000 patients and survivors, data from 12,000 audiograms from 2,400 patients and survivors, 1,400 DNA samples, and 1,600 serum samples. We had 8 work packages comprising the data center at number 1 and the project management at number 8, with number 7, dissemination exploitation, headed by Kylie O'Brien. After that, the topics were covered by the data, by the work packages: fertility, genetics of fertility impairment and hearing loss, ototoxicity, and health-related quality of life. So I'll go through some of the papers that have been published since our first. We have had about 21 papers covering management, methods and protocols, guidelines, and results of statistical and biological analysis. And as is true for PanCare SIOP, more papers are in progress. You can get a list of the publications from the PanCare office by emailing info@pancare.eu. So I'm going to start with the Fertility Guidelines, and we've completed all of the objectives of this work package. And there are 4 papers. 3 of them were published as a series in Lancet Oncology, headed by Renee Mulder and Anna Font-Gonzalez. And these are huge achievements. They involved a large number of people from all over the world, with an extraordinary number of telephone calls to describe fertility preservation for female patients, for male patients. And then we thought that the ethical considerations and communication considerations were deserving of another paper. So we also had a fertility intervention, and I'll just mention this briefly. This was led by Anna Borgman-Stout and her group. She effectively put together a consortium within a consortium, and she had a lot of data providers who were just contributing data to this work package and didn't have a real presence in the main consortium. But that worked well, and that was a successful achievement. So she found that both knowledge and empowerment were increased after an educational intervention in several European centers. And furthermore, she found that inability to recall fertility counseling was associated with lower use of cryopreservation. So this whole idea of counseling and providing education to patients and their families has to be accompanied by some effort to make sure people really do hear what you're saying and really do understand it. And perhaps you have to do it again, as was said earlier. So fertility impairment was a large observational study, which comprised a cohort study, and 10,000, nearly 11,000 women were invited, and 60% participated. Overall, 17% had some form of fertility impairment, and this was defined broadly. This is a paper published in last year, 2021, by Marlene van den Berg, describing treatment-related fertility impairments. In the case control part of this study, she found among 450 cases who were women with fertility impairment, compared to 882 matched controls who were survivors, female survivors without fertility impairment, that there were strong associations with fertility impairment And the cyclophosphamide equivalent dose was one of the main outcomes, and this is the total alkylating agent exposure. So this paper was mainly concerned with establishing a dose response effect and a threshold effect. And indeed, they found that there was an odds ratio of 2.6 for the overall CED score with a dose response effect. Other agents that were involved excuse me, in fertility impairment included these listed below, as well as the usual suspects, lower abdominal radiotherapy and TBI. Mary van den Heuvel-Eybrink was the leader of a group that looked at the effect of genetic variation. And in this case, anti-Müllerian hormone was used as a proxy for ovarian damage. And these collaborators found that this SNP, which detoxifies alkylating agents in the liver, was significantly associated with lower AMH levels. In contrast, another SNP showed a significant protective interaction effect. These are very complicated studies to interpret, and I would say that we're probably only at the beginning of our understanding of the genetic underpinning of the interaction between cancer treatments and human biology. So we have a lot to learn here. Looking at the genetics of hearing loss, we also found that there were SNPs associated with hearing loss in non-cranial irradiated cisplatin treated children with cancer. This project was focusing on the effect of cisplatin to induce hearing loss in children, as opposed to carboplatin. And we found, and I'm not showing this, but we're coming to, we will publish, hope this year, the finding that approximately 50% of children who are treated with cisplatin had significant hearing loss, which is an enormous problem for them and their families. So 14 variants in 11 candidate genes were genotyped, and we're looking at a resource here, as Lars said. This is a huge resource, both for the participants in PanCare Life, but also for other investigators who would like to use these resources in the future. Health-related quality of life, we're just at the start of publishing from this material, and we will be seeing more papers from this work package in the future. We developed a policy on access to the PanCare Life data, and as with PanCare SIOP, these data are embargoed for 5 years to allow the principal investigators to complete the analyses that they outlined at the start of the project. So people get 5 years after the end of the project to finish all the papers that they said they would finish before the data is made available, I would say, to external researchers. The external researchers will still have to be to work with the management team and the executive board going forward. And as with PanCare SIOP, there is an application of intent form that people are asked to complete. So I'm immensely grateful to survivors and their families for their participation in this project, to all the data providers for working so hard to deliver all these data, to all the PanCare Life members for friendly and open cooperation, to the work package leaders who allowed me to present the work package specific results, to the European Commission, without whom this would never have happened, and all the other agencies who contributed funding, and to you, the audience, for your attention, and I'm happy to take questions. Thank you.